Okay. Hello, everyone, and welcome to the section Pathogenesis Immunology 2, Part 2. And Dr. Yvelle Hayes and myself will co-moderate this session. And yesterday, and Pathogenesis and Immunology Part 1, I think mainly focused on the virus and cellular reactions to try to explore the pathogenesis and immunology related to the FMD infection. And in today's session, part 2, and I think we are more focused on the human immune response to the FMD infection in the host, in natural host. And as we know, the human immune response plays a very important role in the protection against FMD infection in the host. And as we all know, and FMD is antigenically variable, and also the vaccination only induce a short-lived immune response, and also with the co-infection with a different serotype of FMD also can occur in the field. So in order to cope with this unique characteristics of the FMD infection or vaccination, I think it's important for us to understand immunological mechanism for how the cross protection can be induced by the vaccine in order to deal with the variable FMD isolates or genetically variable. Also, we know the why. Also, try to understand why how long-lived antibody response was induced by the natural infection. Because when you compare it to the vaccine-induced immunity, natural infection normally can induce quite long-lived immune response. And also, it's also very important to understand what's the impact of co-infections on the immunology, on the pathogenesis, or immunology response in the host. So I think this such knowledge is quite it's essential for us to develop the better preventative tools and the vaccines to effectively control the FMD globally. So I think in today's sessions, so we're going to have 4 excellent talks with the updated progress on this aspect. I think without wasting any time, I think I'll ask my co-moderator, Dr. Leo Hayes, to introduce our invited speaker first.
Thank you very much, Zidong. It's my pleasure to introduce the first speaker of today in this session. It's Dr. Mariano Perez-Filguare. Dr. Perez completed his PhD working on experimental FMD vaccines at the University of Buenos Aires. After that, he completed postdocs at the University of Lincoln in the US, as well as working as a junior researcher at CISA in Spain. On his return to Argentina, he joined the National Institute for Agricultural Technology and the National Council for Scientific Research. He is currently the coordinator for the National Project on Animal Pathogenesis and Immunity at INTA, as well as the principal investigator at the CONICET project. He's also the deputy director of the Institute of Virology and Technology Innovations. His research group focuses on the protection afforded by FMD vaccines in natural hosts, focusing specifically on the mechanism of cross-protection among FMD strains, which is the topic of his talk today. With that, we look forward to his talk and hand over to him. Thank you very much.
Hello, everyone, and thank you for being here. Today we are going to talk about vaccines. The title of this presentation is Vaccine-Induced Immune Mechanisms Underlying Cross-Protection This work has been done at the Institute of Virology at INTA, Argentina. As you know, the level of specific antibodies against the virus are usually related to the chance to result protected against an infection with the homologous virus. This is a result, at least in Argentina and in other countries, of thousands of experiments where they related the challenge experiments with the antibody levels that they could measure by different techniques. In this case, we are talking about the PPG done in Argentina, the percentage of animals that could be positive for this test, and the liquid phase blocking ELISA titers. The challenge experiments in this case were performed on 90 days and were correlated with the ELISA titers taken at 60 days. That gives you a hint, a number that helps you as a reference to quantify the herd immunity and see if the level of the population immunity is good enough to cope with any potential infection that may happen in the herd. This is not exactly the same with the heterologous strains, and the strains that many times circulate in the field are not the same that are included in the vaccine. Here just a few of hundreds of examples where field strains really overpass the protection and the immunity induced by vaccines. Many of them are in our region, but some of them are in Southeast Asia, Africa, and based on that, different groups, they have developed different strategies to cope with this problem, and somehow they prove to increase the payload, to test different strains other than the original ones, or sometimes mix more than one strain. So the OIE has recommended the use of index, the R1 index, that would tell you using serology how similar or different is a field strain from a vaccine strain. We have been talking about this during the first day, but it has a number of problems. One of these is that this R1 value varies according to the homologous serum, if it is strong or not, and that will depend on the vaccine, what is the payload, the adjuvant they have used to produce this sera. It's really hard to harmonize between labs, even when you are using the same reagents. And also the sera is always taken at a fixed time, but we know that during the time the sera changes in terms that the immunity changes, so it can vary according to the time. This has been reflected in a paper by Brem and et al. From Berha's laboratory, where they have performed using the serotype A, different serotype A strains, a number of heterologous challenges. They have seen that in many cases, groups with high neutralizing antibodies titers do not always correlate with heterologous protection, as you can see here. But also they have seen that in some cases, animals with low neutralizing antibody titers were protected. It was like a random situation. It didn't depend on the level of neutralizing antibody. That triggered a number of strategies to try to fix this problem, but very few have really been focused on what are the mechanisms behind this kind of problem and behind this kind of results that they are seeing in the experiments. We have developed this a few years ago, this experiment where 4 groups of animals, of cattle, were vaccinated following different schedules. 3 of them with the monovalent vaccine A24, one with a low payload, 10 micrograms, one with a high payload, 40 micrograms, the third one with 2 doses of a lower payload vaccine, and the fourth one was trivalent vaccine carrying the same amount as the high payload. And we use as a challenge strain the A Argentina 2001 that we knew from our previous experience that does not was not protected very well by the A24 Cruzeiro vaccines. So 30 days after the primary vaccinations, animals will challenge, and the results are these: the low payload, only 60% of the animals, 2 of them were suffer a generalization of disease. 1 in the high payload monovalent vaccine, that means 80% of efficacy, and the other 2 were 100%. None of the animals show any kind of lesions in the case that they received 2 doses or they received the trivalent vaccine. Of course, both naive animals were not protected after the challenge. This is also expressed in the clinical score, where the highest scores belong to the naive animals, because the score rises really fast and reaches values that are the highest of all of them. From the non-protected animals, the non-protected animals show a slower progression of the score and also lower values, but all of them, they have lesions in the limbs. There was a third group that they really didn't show blisters, but sometime during the period, the 7-day period, they present some kind of transient hyperthermia, and all of them, they belong to the lowest payload vaccine, the 10 micrograms per doses. Of course, we do a battery of serological tests and cellular tests, and we calculated the R1 based both on liquid phase blocking ELISA and on BNT. As you can see, many of the R1 values did not correlate with the results obtained in the challenge experiment. Actually, they were very, very low, even when the level of homologous sera was very high. That was one of the recommendations when we were trying to set up a reliable system based on R1. But anyway, the most 5 out of 8 they really failed to predict that, and 1 was really borderline. So that said, if we study the titers against the 4 FMDB strains in each of the experimental groups, you can see that there are really significant differences in the A24 Cruzeiro group, but these differences disappear mostly for the A Argentina 2001, because actually these antibodies represent between 5 and 6% of the total of the A24, so that's just a fraction. One thing that is also interesting, that may be also related to the kind of assay, is that in both cases, in even when the animals did not receive any strain C3 indial or one campus, all of the monovalent vaccines, they have antibodies against these two strains. So more or less the same was what we observed in the neutralizing antibodies, and most of the significant differences were lost when considered the A21. Then we decided to compare not by group but between protected and non-protected animals. In this case, we use all of this, the total antibody neutralizing, the interferon gamma, also a cellular test, IgG 1, 2, and M, and in none of these cases we found differences between the non-protected and the protected animals. So that was not really the reason why these animals suffer a different fate after challenge. One of the parameters that really show difference was a quality one that show the avidity index against the challenge strain. In that experiment, we could see that protected animals were much higher than non-protected animals, and that also was reflected in this kind of ladder that is on the left, where you can see that the three non-protected cattle are the first three animals that are in this kind of scale. But it was very interesting that this difference was also observed when the avidity index was measured against two other strains, and one of them was from a different serotype, the O1 campus, and the protected animals, once again, they were much higher than the non-protected animals. That led us to the idea that it could be some kind of correlation between the values of the avidity index that were observed against one strain and another strain. So that's what was our next experiment. And this is what we found. We found that in all the cases, the animals have very similar avidity index against different strains. That means against A24 or A2001 or one campus. That was reflected by these strong correlation curves that we have seen. Of course, there are a number of potential interpretations of these results, but does it really mean that these high affinity epitopes are somehow preserved in different strains, even in different serotypes? Of course, this is a limited example because this is only 3 strains and only 2 serotypes, but this can be tested in other, and that could be a chance. So just to end, I will just comment on an experiment that it's being now performed in Argentina in a collaboration between INTA and Plamilan, and where we are trying to understand this problem from 2 points of view. One of them regarding what happened in the animal when you immunize these animals with different vaccine schedules. That means different formulations and different amount of immunizations. But at the same time, we will test this with a set of chimeric viruses that will go between a vaccine strain, that is the O1 campus, and a field strain, O Ecuador, that during 2009 and 11 really was a problem in South America, because did not fit very well with the O1 campus, and it was really a failure of these vaccines. And further experiments demonstrated that there was not really any kind of cross protection between these 2 strains. So we will generate by progressive mutations a number of chimeras that will be tested by neutralization against the different sera obtained a long time and using different vaccine schedules. So as a conclusion, we can say that just considering total neutralizing antibodies before the challenge may not fully explain the protection against the heterologous challenge in vivo. The protection against infection by a virus strain that is heterologous seems to be linked to the ability of the sera at the time of challenge, and the high correlation of the ability indexes of each sera, even when the vaccine schedule received is different, or if you use different virus strains, that may suggest that the epitopes recognized by these high ability antibodies may be mostly coincident, and probably they are preserved among these three strains. So just to thank you, I will show you the staff that has been working on this, a lot of people, especially Sebastián Di Giacomo, that is the PhD that carry most of our work. But of course, we have been able to do this experiment with the support of Pathogenesis that generates the experimental vaccines, Senasa, that took care of all the safety issues related to carrying this kind of experiment, and people from the USDA that is working right now with our group in the new project. I thank you very much, and I listen for your question if you have any. Thank you very much.
Thank you very much, Mariano, for the nice talk. And in this section, we have three selected talks. And the first talk is the foot-and-mouth disease virus, localization on follicular dendritic cells, and the sustained induction of neutralization antibody is dependent on the binding to the complement receptor, CR2/CR1, which will be given by Lucy Gordon. Lucy is a final year student at the Purbright Institute under Dr. Harris and Professor Brian Charleston supervision. And Dr. Ruben Marrero Desti Venegas will be the next, further with his work on the fundamental characterization and molecular modeling of site A specific IgG and IgM neutralizing antibody. Ruben is the microbiologist and worked at IAMIBO and Inter, focusing on the interface between NGS-derived data and structure bioinformatics as resources of information for vaccine design. Dr. Carolina Stingfield will finally present her work spontaneously and staggered foot-and-mouth disease co-infection cattle. Carolina is a veterinarian and who did her PhD study on pathogenesis of FM persistent and the Technical University Denmark, and now currently work at USD Aris and Plum, with primarily focus on the study pathogenesis and transmission of FMD in the cattle. So please have your question ready for discussion after completion of this talk. And invited speaker and three presenters will be available for your discussion. Thank you.
The title of my talk is Foot-and-mouth disease virus localization on follicular dendritic cells and sustained induction of neutralizing antibodies is dependent on binding to complement receptors 1 and 2. So one of the features of foot-and-mouth disease virus infection, which has a major impact on the control and eradication of foot-and-mouth disease, is the existence of the carrier state. Only ruminants are considered FMDV carriers, and among them, around 70% of African buffalo become carriers after acute infection and can carry FMDV for up to 5 years or more, which is why African buffalo are considered the primary reservoir of FMDV in Africa. Over 50% of cattle exposed to FMDV become carriers, and although current vaccines prevent clinical disease, they do not prevent primary infection of the nasopharynx. Therefore, vaccinated animals can still become persistently infected carriers of FMD. Another fundamental problem, which remains to be understood, is the short duration of immunity after vaccination, which contrasts with the prolonged immunity after natural infection. Our group has previously shown that FMDV locates rapidly to and is maintained in the light zone of germinal centres following primary infection of naive cattle for up to 38 days post-infection. The hypothesis was that FMDV immune complexes trapped by follicular dendritic cells, or FDCs, may represent a possible source of infectious material and may be important for maintaining the high titers of neutralizing antibodies seen post-infection. So a little about FDCs. They are found in B cell follicles of germinal centres in secondary lymphoid tissues. They are necessary for germinal centre formation and, together with B cells, are crucial for an effective humoral immune response. They have this unique ability to trap antigen-antibody immune complexes on their surface for long periods of time, and a number of different pathogens have been associated with this FDC retention, including but not limited to prions, human and feline immunodeficiency virus, and bovine viral diarrhea virus. So previous work at Pirbright has established the mouse model for FMDV persistence. They showed that FDCs present in the mouse models had the ability to trap and maintain FMDV in the splenic light zone germinal centre for up to 63 days post-infection. So the aim of my experiment was to assess FMDV antigen retention on FDCs and the generation of the specific immune response in vivo in mice treated with a complement receptor block. So here is a schematic of an FDC, and in this teal-like colour, these are complement receptors 1 and 2, and they are responsible for trapping immune complexes presented predominantly via germinal centre B cells. So the design of the study was to inoculate mice with an anti-complement receptor monoclonal antibody named 4B2, and then the following day infect animals with FMDV, and then look at the ability of FDCs to trap FMDV up to 28 days post-infection. A similar study done previously by Ho et al. Used a commercial rat anti-complement receptor 1 and 2 monoclonal antibody to block the binding of HIV in vivo in mice. However, as we were looking at long-term persistence of FMDV, we used this mouse monoclonal antibody 4B2, which has been shown to block complement receptor for up to 6 weeks in vivo in mice. So I'll run through some of our key findings. So firstly, the viremia. By 2 days after infection, a statistically significant 10-fold increase in viremia in sera was detected in the monoclonal antibody 4B2 group compared to the isotype control group. So on the left, this is via plaque assay, and on the right, qPCR. So by 7 days post-infection, the viremia was cleared in both groups, and no detectable virus was detected by plaque assay or PCR. So as I said, IgG1, that was our isotype control group, and PAP, which is peroxidase, anti-peroxidase, this was our positive control, as we know it binds to FDCs via complement receptors 1 and 2. So here you can see very clearly positive staining for PAP in the FDC network. These are marginal zone macrophages which line the light zone germinal centre, and here is the merge with the blue as the nuclei. So when you pre-treat with 4B2, PAP is unable to bind in the FDC network, and this was proving that the 4B2 monoclonal antibody was working in the mice in vivo. So then with that isotype control, after infection with FMDV, we were able to show FMDV binding in the FDC network. However, pre-inoculation with 4B2 prevented FMDV binding in the FDC network. So this is a graphical representation of the confocal data. So in blue we have the 4B2 and in the red squares are the isotype control group. And you can see from 7 days post-infection up to 28 days post-infection, there is a very clear effect of pre-treatment with the complement receptor block, preventing FMTV antigen retention on FTCs. I've highlighted some mice in green, which are highlighted throughout the presentation, and these are mice where the antibody treatment did not appear to be successful. This was not too surprising, and it's also why we used 8 mice per group per time point. This table is the same data which is represented on the graph, just showing in the top row how many germinal centers were imaged per group per time point, and then the percentage, the mean percentage of positive germinal centers for FMTV. So these results indicated that FMTV was binding to FTCs via the complement receptors 1 and 2. Further investigation using super-resolution microscopy showed significant co-localization of FMTV antigen with complement receptor positive FTCs in the spleen. And then the ImageJ software confirmed a highly significant and preferential association of the FMTV antigen with complement receptors 1 and 2 on FTCs when compared to the null hypothesis that the pixels were randomly distributed. So then I confirmed the confocal data using PCR, determining the presence of FMTV in the spleens. So from 7 days post-infection, you can again see the clear effect when 4B2 has blocked the complement receptors in vivo. FMTV is not retained on FTCs. And these results again indicate that FMTV is binding to FTCs in the germinal centers via complement receptors 1 and 2. Next, I looked at the virus neutralizing antibodies. So you can see at 7 days post-infection, both groups have mounted an immune response to FMTV, and by 14 days post-infection, there is a significant difference between the groups. And this lasts up to 28 days post-infection, which was our final time point. So mice treated with the complement receptor block generate a lower titer of neutralizing antibodies against FMTV. And this data suggests that FMTV needs to bind to FTCs via the complement receptors 1 and 2 in order to produce an effective neutralizing antibody response to FMTV. So despite the differences in titers of neutralizing antibodies, there were no differences in the FMTV-specific IgG on the left or the IgM on the right. So therefore, I looked into the specific antibodies and their affinity to FMD virus-like particles to determine where the differences in the neutralizing antibodies lay. So here on the left are the total IgG to the FMDV GH loop. You can see, again, at 14 days post-infection, there's a significant decrease in antibody titers to the GH loop and mice treated with the complement receptor block. And again, this lasts up to 28 days post-infection. So it's been established that in mice, the GH loop is a neutralizing epitope inducing protection against FMDV. So I also looked at the avidity of the antibodies and determined that the virus-specific antibody induced after the complement receptor blockade had reduced avidity to FMD VLPs. And you can see again, starting at 14 days post-infection. So FMDV needs to bind to FTCs to produce high avidity neutralizing antibodies to FMDV. So just to summarize, complement receptors 1 and 2 are responsible for the binding and persistence of FMDV on FTCs and germinal centers, which are essential for maintaining neutralizing antibodies with a high avidity to FMDV in mice. And I'd just like to thank my supervisors, Eva and Brian from Pirbright and Neil Mabbott from Roslin. Also the Animal Services, Bioimaging, Viral Immunology and Molecular Virology group at Pirbright. Ludmila Kulik for providing the monoclonal antibody 4B2, and Nick Julef for his previous work on the FTCs. Thank you.
Hi, my name is Ruben. I work in Yavimo, Argentina. I present my work, functional characterization and molecular modeling of Cy-A specific IgG and IgM neutralizing antibodies. As a background, we can see here the viral interest that is mediated by virus interaction here that is occluded by the second kind of interaction between antibodies in gray with the viral capsid. site, the barcodes have different antigenic site. Here the antigenic site are at the GH loop of the PP1, where there are the motif RGD that is the main target of neutralizing antibodies. So considering that antibodies are key molecules for FNDB control, but there are few molecular structure data and information. Specifically here we have a PDB a database of PDB entries for monoclonal antibodies, and we find only 5 entries with a 20-year difference, which means an issue. So, considering several publications, we define our long-term goal by a deeper knowledge about the molecular structure of the induced antibody repertoire could benefit FNDB vaccine design. And our current goal is model and characterize the molecular structure and functionality of novel FNDB monoclonal antibodies. Considering our design, we first will select our input, our antibodies, then conduct several experiments that find antigenic site mapping by ELISA, clone the antibodies model, and also model the antibody interaction. So looking for our input, we select monoclonal antibody for serotype A strain, specifically A24 Cruzeiro, described and obtained at Ceban in Argentina. Those antibodies you can see here, the table of the paper, display different isotypes, those IgG and IgM here, but also contracted interaction pattern with several FNDB strains, and both antibodies recognize epitopes at the antigenic site as of BP1 that was only mapped in the second paper by Ceban lab. And as a result, we first implement a peptide ELISA, a competitive ELISA for a better epitope mapping at say A, and we designed 15 peptides with point mutations. Here's our wild type in green, and here is the RGD motif, and the single point mutants, and there are colors that mean the relative interaction index, where red means a higher index or a worse mutation. And here is a resume of that, where in blue we have the IgG that displayed a more sensitive pattern of the spike prime of RGD. And both might recognize overlap of the epitopes around RGD motif, but IgG is less tolerant to mutation at that point, as I say. But the IgN epitope is wider with sensitive residues at the periphery. That part. Here, the molecular modeling pipeline is a graphical representation with several steps, and in general, we define a funnel design with a water input software and protocols that convert infused solution, interaction solutions. Going to the molecular cloning, here is the germline, the genome that codes for the H chain of the antibodies that then convert in a transcript. Here is where we amplify the sequence by RT-PCR. Here the CDR that determine the complementary determinant regions that determine the paratope, the antibody binding regions in the antibody properly. So here we display some information about the new antibodies. We always set a comparison with the reference antibody of serotype C, that's antibody for serotype A. And here you can see that for CDR1 and 2, they display the same length, but the CDRH3 is different, and 44 have more amino acid, but the other are smaller. You can see here in blue for the IgG, and in yellow a bigger CDR for the reference antibody. And here we also put the second antibody in pink, and here the alignment for the L chain, and here the analysis for the L chain, And again, all the CDR display the same length, but not for the L1 CDR. That in conclusion, the map 1A12 was the smallest of the whole group. Nevertheless, the interaction probability was not affected. Here we can see an interaction probability determination, and in red we have the information for the IgN that compare with the interaction probability calculated for the reference 44 map, and we can see that the interaction probability remains. And next we advance to the antibody modeling, and we use 3 different flavors, different software, and obtain our models again compared with the 4C4 reference antibody, and in green it means that the mod probability score is good for that reference, and we have the same green models for our 2 antibodies. We advance for the antigen-antibody docking stage. We use again the reference 4C4 antigen-antibody complex of reference to recapitulate the obtaining of neonative docking solution. Here we represent 3 different experiments with different conditions to recapitulate. Here we obtain the characteristic funnel pattern with a lowering energy across different solutions that reflects here. In red there are the reference 4C4 peptide inserted interacting with the antibody, and in blue we can see a recapitulated solution near the native structure, and here a gray here a more structural distant solution. Next, we applied those protocols to our antibodies, and again here we can see that we obtained the funnel pattern, the energy funnel pattern solution, but we advanced to ELISA assisted scoring. Finally, we further scored the docking solution with functional ELISA information. Here we take that solution and calculate every single point mutation. I mean computational calculate data, determine the energy interaction. We determine a profile B and profile I emerging for the ELISA. We calculate the coefficient of determination between again both profiles that became in our functional score, and that point out our final candidate solution here. And here you can see the top docking solutions, and this is the IgM in red and the IgG in blue. This is the peptide, and you can see that the peptide here has a flatter interaction with the map, unlike the interaction with the other antibody. With the peptide will be more insert in a more concave paratope. I mean, if we compare with the ELISA, here you can see that will make sense here in red that kind of interaction, in blue those that means maybe a more deep if unfocused in certain regions. As a conclusion, we test a computational protocol guided by ELISA functional information and obtain candidate molecular model of novel antigen-antibody interactions. The protocol yields relevant antibody models with differential paratopes and antigen interaction topologies in concordance with their isotype classes. And last, we demonstrate that functional prediction techniques can be applied for the study of modeling of interaction phenomena between FMDB and antigenic sites. Thank you for your attention.
I'm Carolina Stanfeld, and I'm going to talk about a study of FMDB co-infection in cattle that was recently done at the Plum Island Animal Disease Center. This first slide here is a brief reminder of FMDB pathogenesis in naive and vaccinated or otherwise clinically protected cattle. In cattle, FMDB infection initiates in the nasopharyngeal mucosa of the upper respiratory tract. And after establishment of the primary infection, there's a phase of systemic generalization with viremia and clinical disease in naive animals. Meanwhile, in the clinically protected animals, the virus remains restricted to the upper respiratory tract through the phase that we like to refer to as neoteric subclinical infection. And the reason that we call it by that name is to differentiate it from what comes later on in a subset of animal, which is the persistent phase of infection, during which the virus is also restricted to the upper respiratory tract. The main difference between this neoteric and the persistent subclinical infection is that during the neoteric phase, the animals are usually shedding more virus in oral and nasal secretions compared to what they do during the persistent phase. So the main topic of this presentation is FNDV co-infections, which is a somewhat common finding during field samplings of both cattle and buffalo. That means that it's not uncommon to detect more than one virus strain in the same animal. However, the finer details of how these multiple infections have come to be, especially in relation to each other, have not been previously examined. So we set out to design an experimental study to answer some of the questions we had in relation to FNDV co-infection. We specifically wanted to investigate the coexistence of multiple strains within the same animal. Additionally, we wanted to see how being persistently infected with one virus strain would affect subsequent infection by a heterologous virus. And we also wanted to investigate if and when these viruses might recombine. Our study included 4 groups of cattle. The first group was simultaneously infected with FNDV 824 and O1 Melissa through intranasopharyngeal inoculation. The second and third groups were initially infected with just the 824 virus and then superinfected with the O1 Melissa virus 21 days later. And finally, the fourth group was also infected with the 824 virus first and then superinfected at 35 days. And all animals were monitored through 4 or 5 weeks after the final infection. I'm going to walk through the main findings in these study groups, starting with the simultaneously infected group number 1. This image illustrates a timeline of strain-specific detection of the 2 different viruses in serum, nasal swabs, and OPF, or PREBANG samples, for the 4 individuals in group number 1. I'll show images of a similar layout for the subsequent groups. And in all of these images, detection of A24 is illustrated in blue, while O1 Melissa detection is depicted in red. The purple lines represent the period of clinical FMD. For the PREBANG samples, which represent most of the detection through the later phases of the study, so the right-hand side of the image, there are also some light blue and pink lines which indicate that the samples were positive for the indicated virus by strain-specific PCR, but negative for that virus by virus isolation. And that typically occurred, as you might see, towards the tail end of detection for that specific virus. So in short, all of these animals were infected by both viruses and they did develop clinical FMD. They were also all persistently infected. 3 maintained infection with the A24 throughout the study period while clearing O1 Melissa, while the 4th animal was persistently infected with O1 Melissa. We harvested samples of vesicles during the clinical phase of infection from the first study group, which is illustrated in this image. Again, the A24 is in blue and the O1 Melissa is in red. And in short, 3 of the 4 animals had vesicles containing both viruses, either separate or together, while the 4th animal only had vesicle caused by A24. That brings us to study groups 2 and 3, which were superinfected with O1 Melissa 21 days after the initial A24 infection. This is a similar layout as I showed before, showing strain-specific PCR detection in samples collected from the 8 animals in group 2 and 3 through the first 21 days. All of these animals were infected after the first inoculation. They were also all positive for A24 in PREBANG samples. However, when we superinfected these cohorts with O1 Melissa at 21 days, only 2 of the 8 animals developed clinical FMD, as illustrated by these purple fields. We did, however, detect O1 Melissa in nasal swabs from some of the superinfected animals that remained subclinical. When we move further on in time, things get a little more exciting, as when we sequenced the viruses recovered from the PREBANG samples by NGS, we found dominant, as in consensus-level interserotypic recombinant genomes in consecutive PREBANG samples from 4 of the 8 animals. And similar as the first study group, all animals maintained persistent infection by only one virus. So 4 of the animals were persistently infected with the A24, 3 maintained O1 melissa infection, while 1 animal was persistently infected with a recombinant virus. The 4th and final group was infected with A24 and then superinfected with O1 melissa at 35 days post the first infection. Similar as previous groups, all animals were infected by the first exposure, and they were also all positive for A24 in probank samples. But in contrast to the previous group, all animals in this group developed clinical FMD when superinfected with O1 melissa at 35 days. We did detect a dominant recombinant virus in 1 OPF sample from a single animal. However, none of the animals in this group maintained persistent infection with either of the viruses through the study period, which was a little bit unexpected. And you can see there was some intermittent detection in 1 of the animals, but that one was VI negative towards the end of the study, suggesting that it was likely in the process of clearing infection. This is just a brief overview of the consensus level recombinant genomes that we recovered from the OPF samples, and you will hear more details about these recombinant viruses in a subsequent presentation by Ian Fish. But just briefly, in the top of this image, these genomes are aligned to O1 melissa, and in the bottom portion they are aligned to an A24 reference, and the black bars in the alignment indicate that the genomes are different from the reference that they are aligned to. So the short version here is that all of these recombinants had capsid coding regions derived from the O1 melissa virus and non-structural coding regions derived from the A24 virus. And in some cases, as you can see for the animal that is circled in yellow, there was also evidence of multiple recombinations events. And interestingly, that one individual was also the one animal in which the chimeric virus persisted throughout the study period. So to summarize the findings, we experimentally reproduced heterologous co-infection through either simultaneous or staggered exposure to 2 distinct FMDVs. We did not detect any recombination in vesicles from the simultaneously infected animals. That clearly doesn't mean that there's no recombination going on at a molecular level, but it did not result in any recombinant genomes at a level where we could detect them using Illumina sequencing. Heterologous superinfection mediated partial clinical protection when exposure was separated by 21 days, but this protection was overcome when we separated the exposures by 35 days. Interestingly, separating the exposures by 35 days seemed to also mitigate FNDV persistence. We did detect interserotypic recombinants in consecutive OPF samples from multiple of these super-infected animals. We're quite intrigued by this detection of the dominant recombinants and the super-infected carriers, and we are hoping to be able to do some more work to investigate this. Specifically, there seemed to be a selective advantage of the capsid coding region of the virus from the most recent exposure, so the O1 melissa in this case, and that might be explained by the lack of neutralizing antibodies to that virus during the very early phase of the superinfection. But we'd clearly like to know what happens if we were to swap the order of the virus exposures. The repeated finding of these recombinants in these superinfected carriers raises a question of whether persistently infected animals may function as a sort of mixing vessel and potential source of recombinant foot-and-mouth disease viruses out in the field. We saw that these superinfected animals were shedding some virus during this neoteric phase of superinfection. We don't know if they're actually able to transmit infection at that stage, but it is an intriguing concept. Finally, all of this work was done in Jonathan Art's lab at Plum Island with contribution from a whole lot of people, and we hope to be able to continue some of this work in the future. And with that, I would like to thank you all for listening.
Thank you very much to all of the authors. That was an intriguing presentation from everybody and brought up a lot of questions. There were a couple of questions on the screen, so I'll take us through some of those and then I'll give Zedong a chance to ask some of the questions later as well, or transmit some of them to us. The first questions to our invited speaker, Mariano. Let me just quickly get to this. Aldo asked 3 out of 5, 4 out of 5, and 5 out of 5 is not significant differences in protection, which is as a Fisher exact number of 0.7. So protection is similar due to the low power of the design. Would it have been better to use a PD50 format to quantify protection, 15 cattle? I'm sure we're all familiar with the PD50. And why did you choose to go with a PGP format?
Well, thank you, Aldo, and you're right. We are limited in the number of animals per group. Anyway, we have consultation previously, but we choose the PGP because that allow us to have different groups of animals with different vaccine schedules. The PD50 system considers 15 animals in a single group. Though the goal of the experiment was completely different, we showed that only measuring total or neutralizing antibody may not tell you how good the vaccine may perform against a heterologous strain. And that is because it is measured before the challenge. And what is important is what happened after the challenge, because the memory response triggered by the infected virus after infection is critical for protection. That is not taken into account when you measure that before. So that was, and if you see the groups, we are not stressing the differences in the groups, and when we make all the groups together, we cannot find a difference between protected and non-protected unless we employ a parameter that might give you some hint about the maturation of immune response that we understand that is critical for protection. Thank you, Aldo, for your question anyway.
Thank you very much for the answer. I think all of us working on select agents have experienced the difficulty with working with large numbers of animals, so definitely understandable. There's another question from Toby. How can we identify the conserved epitopes that induces high avidity antibodies for multiple serotypes? Mariano, I don't know if you got that.
Can you repeat it, please?
Sure, no problem.
Yeah.
How can we identify the conserved epitopes which induce high avidity antibody protective for multiple serotypes? It's quite a difficult question.
The short reply is I don't know, but it is critical to map these epitopes. What we have shown is that they may exist. It may exist epitopes that are conserved among strains that where the high affinity antibodies are targeted. So there are a number of new technologies, even in a poster from Perbright, where they are showing new alternatives to measure maturation, and even what Ruben was showing about the paratopes could be somehow changed to map this kind of important epitopes that we believe that may exist among different strains. So I don't know but the technology probably will give you an answer soon.
Thank you very much. Lucy, we're going to come to you next. There was a question again from Aldo. Did I understand it correctly that replication of the FMD and the MAB treated mice was lower? Lower replication could be an explanation for lower immune responses. Old studies with oral infections of pigs show pig without clinical disease that developed FMDV titers. The low antibody response, is it due to low replication or to CR binding?
Yeah, thanks for the question. So I was showing the presence of FMDV antigen and not the replication. It's not clear where FMDV replicates in mice, but it's not being shown to replicate in the germinal center. One paper's shown it to replicate in the pancreas, but otherwise nowhere else in the mice. So we believe it's due to the complement receptor binding.
Thank you. There's another question for you. Does it mean that the virus have to be an immune complex to get into the FDC? Did you check that by isolation of the FDC and splenocytes or in the spleens? And then C1 is involved in classical pathway of the complement system and need antibody bound to pathogen to activate the pathway.
Yeah, again, really good question. Thank you. So we believe that FMDV would be in the form of an immune complex to bind to the FDCs. There are many difficulties with isolating FDCs. So instead I've done, which I haven't shown today, ex vivo immune complex deposition studies where you add different antigen onto spleen sections. So I added immune complex virus as well as just FMTV antigen alone, and it was only in the form of an immune complex that FMTV was binding to FDCs. So I hope that answers the question.
I think it does. Thank you very much. I think, Carolina, the next questions are for you. We'll start off with Michael's question. Does simultaneous infection mean that you mix the virus preps and then inoculated the animals?
Yes, exactly that way. So we had one titer for each of the virus and then we mixed them together.
Okay. Thank you. And then there was a follow-up question from Leandra. Do you have any immunological data for this?
Yes, no. I mean, I didn't show we have serum neutralization data that showed absolutely no cross-reactivity. We also attempted some more assays during the experiment. That was Faina Diaz-Sansegundo that was in charge of that. And we didn't really find anything that was conclusive or made any sense in relation to explaining any of the variables we were looking at. It was a little bit of a messy data. So we have some, but we don't have much that was actually informative.
Thank you. If I can maybe ask a question from my side, the pattern of recombination that you saw was quite interesting. Do you have any speculative ideas on why all of the recombinants was in the structural protein for the one virus and then non-structural for the other virus?
Well, clearly we don't know for sure. Our hypothesis is that they all had the capsid from the most recent virus, which at the point of recombination, they probably didn't have that much neutralizing antibodies against that capsid. It doesn't really explain why they would recombine and why the non-structural region of the egg virus would be a benefit. So, we'd have to do more studies on that. It's something we really want to look at. But yeah, I agree. It's interesting and a little bit surprising to us too that we had so much of it that was similar. But not identical.
Thanks. A couple of other questions as well from Eva. Did you phenotype the challenge viruses in vitro competition assays?
Yeah, we did do co-infections in vitro, and I don't know if you can go back to the talk by Ian Fish that was yesterday. He actually talked about that. When we recorded these presentations, we assumed that this talk would be before his, but it ended up being the opposite way. So there's some follow-up in that. But short version is they behave very similarly in vitro, both the recombinant as well as both the parent viruses. There wasn't any case of anyone outcompeting the other.
Thank you very much. I'm just quickly scanning. Zurong, do you maybe have any questions that you want to ask? If you could just unmute quickly.
I have a quick question for Lucy. So, which mice did you use for your study? What kind of type?
Biopsy mice.
They're going to die, or they all will be survive after infection?
Yeah, they all survived after.
Okay. How old it is? What's the age?
How old? 12 weeks or something.
Okay.
Okay. I'd have to double check that.
Okay. Okay. No more questions from my side. David?
Thank you. I'm just quickly scrolling through the questions. Carolina, there's a couple of questions more for you. Let me just see. like another one from Michael. Have you tried to recover any of the recombinant viruses either directly or by creating infectious clones?
We do have the viruses. We isolated them and we've used them for some in vitro work. So we have not produced any clones, but we have the actual viruses that we recovered from the ProBank samples.
Subclinical infections after the second infection.
I'm sorry, you fell out for a few seconds there. I couldn't start.
Can you hear me now?
Can you hear me now? A little bit better. My apologies for that. Just another question from Aldu. You show subclinical infection after the second infection. That is interesting, as we once performed tongue inoculations with 3 different strains and saw clinical disease in both cows. Could your inocular method be the cause for the subclinical infection? That's a very interesting question because we've seen similar results in South Africa as well.
Yeah, no, it's entirely possible. We do see differences in pathogenesis depending on the inoculation route. Specifically, if we have very attenuated viruses or mildly attenuated viruses, they can cause disease if we tongue inoculate, but they may not if we use aerosol or nasopharyngeal inoculation. But I mean, then you get back to this discussion, like which is the more realistic scenario? And we use the nasopharyngeal inoculation because we want to study pathogenesis at the site of primary infection, which we have shown through contact challenge studies, to be located in the nasopharynx of cattle. It's different in other species, but that is why we use that system. And we feel if in the absent, like when we're not able to do contact challenge because you need more animals, then we find that the intranasopharyngeal inoculation is the system that best reproduces the anatomical progression of the infection in cattle.
Thank you very much for that. There's a couple of additional comments in the chat section, which I encourage everybody just to look at and to further the discussions. I don't see any further questions for any of our speakers. So I'm going to hand over back to my co-chair for closing the session. We're probably closing a little bit early, but I think we can all do with an extra cup of coffee maybe. And just a reminder for everybody to go to Gather Town for further interaction. So Dom, over to you to close.
Okay, thank you very much, Leo. And also thanks to Dr. Merrill, Persil, Figueroa, and Miss Lucy Gordon, and Dr. Ruben Merrill Desti Verex, and Dr. Carolina Steffes for their formative and interesting presentation. And I think we learned a lot about this updated knowledge about the cross protection induced vaccine, and got some knowledge about why the natural infection can induce long-lived immune response. And also we have a very good new computerized tool to help us to identify some of the good epitope to help us to design the new vaccine, which can still maybe stronger immune response for the new vaccine. And also we understand co-infections can cause some of the potential problem about virus recombination. So that's something quite interesting, and also look forward that we'll have some new progress in the next meetings. So again, and thanks to all the speakers and for their good, excellent presentation. Thanks organizing GFI organized meeting. Organizers give us such a good opportunity to chair these sessions. Thank you very much.
Thank you.
Thank you.